systems that obviously are going to be affected. So keep that in mind as we start to go through the lecture, as we start to talk about things that ramp up the system, the autonomic responses, and the things that slow them down. So with that, let me load up our slides for today. And where I plan on getting to today, most of what we're going to be talking about today, we'll talk about blood, we'll get into the sort of anatomic orientation of the heart, and then next Tuesday, we'll start to focus specifically on a cellular level. We'll look at the makeup of the cells, and then we'll look at how those cells all work together as a tissue layer. We'll start talking about the pressures that are generated within the heart, but we can't do that without doing that anatomical background. So some of this stuff at the latter end of this lecture will be reviewed for you guys, but this is a good opportunity to take the weekend and get familiar with the anatomy and brush up on next week. Because this is one of those lectures, especially the cardiovascular lecture, where we build up. You might see the anatomy, you think, okay, I'm fine on that. You might want to, hopefully you don't skip it. But when you get to the cells, we start talking about the cells, and then we start talking about electrical activity, we start talking about pressures, and then we combine it all together. And I assure you, if you do not keep up, the last half of this lecture is just going to blow right over your head. And that, towards the test, is not the time to study the material. You want to make sure that you're up to date on what you're learning, because I've seen it before, especially when I get to the last, which is one of my favorites, called Wigger's diagram. It puts it all together, and that's where I get the most blank stares. So make sure you keep up. All right, with that, let's get right into our cardiovascular lecture. The very first bit of what we're going to be talking about is, again, the blood. We'll talk about vessels, and then, we're going to talk about blood, we'll talk about the heart, and then the last portion of the cardiovascular lecture will be the vessels. So the heart, as we know, is pretty much just an entire pump. We'll learn more about that. The three components of the cardiovascular system: the blood, the heart, and the blood vessels. I don't really spend that much time in blood, but just enough that you guys get the gist of it. We will revisit the most important structure in blood, which is going to be the red blood cell. We'll talk about that in the vessel lecture quite a bit. So looking at the characteristics of blood, give me a second. Looking at our characteristics of blood, we do have about 5 liters of blood in adults. So that comes out to about 9 pints of blood. When you do donate blood, it's only really 1/9, well, depending, of course, on your body size. If you were to take a look at newborns actually only have about a cup of blood in their system. With that, if you were to take a look at the blood and specifically look at the pH, the alkalinity of the blood, blood has a tendency to be alkaline, meaning it's about 7.35 to 7.45. So again, very narrow window that we want to make sure that we maintain our blood, because that starts to disrupt quite a bit if we start to see blood becoming more acidic, lower than 7.35, a condition that we call if I can write. There we go. Acidosis. There we go. Come on, pen. So acidosis would be less than 7.35. That might be the end of it. All right. Anything above 7.45 would be alkalosis. And that's going to start to seriously affect kidney function. It'll start to affect lung function. It really will start to mess around with the dynamics involved in oxygen release from our red blood cells. So we want to make sure that we still maintain within that. And what we'll learn, especially towards the end of our vessel lecture, when we look at our red blood cells, that we have measures in place in order to maintain that pH. And that we'll learn is going to be our bicarbonate in the blood that will resist any changes in the acid levels. If we were to measure the temperature of the body, temperature of the blood, blood has a tendency to be a little bit warmer. So if our core body temperature is about 37 degrees Celsius, our blood is at about 38 degrees Celsius. Don't know exactly why, but that's just a phenomenon that takes place. If you were to take a look at the composition of blood, and we can measure the composition really by what we call centrifuging, which is the process of spinning the blood in which the heavier sediment sinks to the bottom and the lighter sediment, or let's say the lighter portions of the blood, will flow. And so this separation that takes place in the centrifuge will actually separate what we call the formed elements, of which most of it is going to be red blood cells, which we'll talk about in the next couple slides. The other portion of it's going to be water. So water, we have about 55% water in our blood, and that would be in our plasma. Right in between is what we call a buffy coat, which is going to be our white blood cells and platelets, and again, the heavier stuff, the red blood cells, the big formed elements that you'll see on the bottom. If you were to take a look at the bottom right, there's that misconception that deoxygenated blood, meaning blood that is lower in oxygen content, blood in looking at sort of your subcutaneous veins, people might think that the blood is blue, but it actually isn't. So here we can see arterial blood being really bright, and then here we can see deoxygenated blood. So this is blood that is low in oxygen, actually has a slight purplish tinge, and that's all related to the hemoglobin, which is going to have a heme group, which is iron. Iron being slightly red will interact with oxygen to give it that bright color. If we look at that plasma, which we know is about 55%, the top portion which is all fluid, of that plasma, 92% is water. So you can see 92% water. If you were to take a look at the other things that are dissolved in it, proteins being soluble in blood, we see 7%. And of those proteins, you can see some of the other proteins that are present in here. Albumins, which we already described, being the lipid carrier in blood. Globulins would be your globe-shaped spherical proteins. Fibrinogens being your more fibrous proteins present. Biggest bulk of them will be your albumin for carrying your lipid-soluble hormones. If you were to take a look at the other solutes inside, you'll see electrolytes. Electrolytes would be things such as our charged sodium ions, chloride ions, calcium is a big one, potassium. And naturally, if you were to take a look at not energy drinks, but things like Gatorade, and they talk about electrolytes. That's exactly what's in them. It's just the same salts that are going to be involved, and NaCl would be sodium chloride. We do have potassium chloride, which is another salt. All these things that are going to be involved in ions outside in the extracellular fluid as well as the intracellular fluid, which we already learned. Nutrients would be our vitamins, would be our minerals, glucose, that's present as metabolic substrates. Gases would be O2, CO2, gas exchange. Also a very, very minute amount of nitric oxide, which we already learned is a vasodilator. Regulatory substances would be your hormones. And then other waste products, things that we don't necessarily use, that is eliminated by our urinary system. Now, while the plasma is 55%, the other 45% are going to be the things that sink down to the bottom. So these are going to be our big cells. Red blood cells, white blood cells, as well as platelets. And they are called formed elements because they actually are derived from stem cells. And where are those stem cells for blood located? Actually located right in the bone marrow. So our stem cells start to differentiate, and at no point am I ever going to have you memorize of the other basophils, neutrophils, eosinophils. I just want you guys to be able to see that we produce red blood cells, which is a chunk of it. We also produce our immune system cells in the bone marrow. So keep that in mind about how we get these stem cells, how we get our red blood cells into our bloodstream, especially when we talk in our vessel lecture, or actually even a little bit more when we, yes, in our vessel lecture, that's what we talk about. We see our erythrocytes, which are going to be our red blood cells. You'll see our platelets, and then our 5 types of leukocytes, leukocytes being our white blood cells. If you were to take a look at the biggest chunk of all the formed elements, you would note that red blood cells dominate them all. So in terms of our red blood cells, we do have quite a lot of them. You can see how many red blood cells, about 5 million red blood cells in our blood. If you were to take a look at our platelets, we have about 400,000, so it pales in comparison. And our white blood cells, even smaller fraction of that, about 10,000 white blood cells. So if you were to measure the amount of this pellet by volume, we know it's about 45% of the entire sample. That would, being red blood cells comprising most of the formed elements, or that pellet specifically, would be what's called our hematocrit, and that is the percentage of red blood cells. So how much volume is that of the formed elements? This is going to be our hematocrit, the percentage by volume. Now, in order to stimulate the production of red blood cells, we have what's called erythropoiesis, and we'll learn that this is going to take place for the formation of our red blood cells. So as I said earlier, our red blood cells are called erythrocytes. Erythropoiesis is the formation of it. So how do we stimulate erythropoiesis? How do we stimulate red blood cell production? Well, we get that whenever we are in hypoxic conditions, meaning hypo, low, and hypoxic referring to oxygen. So go up in altitude. You may have heard, if you've gone up to altitude, you have to acclimate to it. And that acclimation process is erythropoiesis taking place, where our kidneys are stimulated to produce a hormone called erythropoietin, also known as EPO. And EPO, erythropoietin, is, like I said, a hormone that's produced. And there are ways, given that our red blood cells are meant to carry oxygen to our active tissues. It's been a phenomenon that's been discovered in ways people would increase their oxygen carrying capacity is through stimulating erythropoiesis. So what people have done in the past, what's called blood doping, athletes would have a tendency to, not all athletes, but those that were trying to get a competitive edge, would take a pint of their own blood, and then they'd wait for erythropoiesis to take place and bring back their normal volume of blood, and then right before the race they would inject that same blood back into their bloodstream. And what that ended up doing was now your red blood cell count was higher, and theoretically you should be able to deliver more oxygen to your blood. However, what that does is, now that you're raising your blood volume by 10%, that's an extra strain on the heart. So naturally, what is a huge side effect with doing something like that is any sort of cardiac event, right? Heart attack or stroke that could take place. Other ways that people have done it would be EPO injections. So they would actually inject the erythropoietin, and that would stimulate red blood cells as well. Obviously, these are things that would get them kicked out if they were found. But
Neil Armstrong, were his medals taken away from him?
Lance Armstrong? Lance Armstrong? Lance Armstrong, I think, because I think he had taken testosterone injections, hadn't he?
No, he blood doped.
Did he?
He did take away his medals.
Yeah, well, it's what people do to get an edge. I actually spent a good amount of time too with our UCLA athletes, and a lot of them got tested. So anybody that actually was diagnosed as an asthmatic had to take a test where to see how much they would respond for the use of Ventolin. Ventolin being an adrenergic, or if they were taking any corticosteroids of any kind. They wanted to make sure that they actually had a valid diagnosis for it. And people have used albuterol to prevent any, or at least to increase the amount of air going into their lungs. So in order for them to actually be allowed to take Ventolin would be a positive diagnosis for some sort of asthma. So there's a lot of particulars that are related to especially sports, regulatory agencies that are there to make sure that whatever medical use you have, it's justified. White blood cells, if you were to see, we have 700 red blood cells to 1 white blood cell, of which we have the 5 different types. I'm not going to really go too much into it because honestly I'm not going to test you on it, but keep in mind that we do have 5 types, of which they all have different functions. And you can see that based off the next slide over are basophils, which would be involved in inflammation. There are 3 neutrophils, eosinophils, and monocytes, which are related to phagocytosis, or have the function for phagocytosis, meaning they basically eat any foreign cells that don't belong. And then lymphocytes, which are all related to a concept called adaptive immunity, which is how we adapt to any what's the word I'm looking for? Any flus or colds or viruses that we do get exposed to. Our red blood cells, again, erythrocytes, you'll note that the features behind them is that they are biconcave, meaning they are literally like a semi-donut shape. And the purpose of this is to actually increase the amount of surface area that's exposed to the bloodstream. So here, if you note, by having a smaller type cell, the diameter of the cell is just enough to fit within a capillary. And in this image to the bottom right, you can see the width of a capillary. The capillary is the smallest vessel that's going to be able to travel nearby to all the nearby cells. And you can see that it literally will fold and fit right through in between. That allows it to not only fit within the capillary, especially our smallest vessels where they can kind of fold like a taco and be able to make their way through. But it also gives it the most surface area to be exposed to so that the hemoglobin that's contained within, and the hemoglobin is a protein that I'll talk about on the next slide, can be exposed to the most gases. And that gives it the ability to increase the surface area. So something I won't really get too much into, but it gives us the highest amount of surface area to volume ratio. We want more surface area exposed for the amount of stuff, the width or the contents of the vessel contained within. The lifespan, if we take a look at red blood cells, do not last forever. So they only last about 120 days. So the question that you'll start to think about, especially towards our vessel lecture, is what happens with red blood cells that die? Do they just die and we just pee them out? Do we get rid of them? No, we need to get them out of the bloodstream, and we need to get new red blood cells that are made back in the bone marrow back into the bloodstream. So we'll learn how, what mechanism is in place, what specific feature of the capillaries are present to allow red blood cells in and out. Why is the lifespan only 120 days? You would specifically see that it doesn't actually, it's been stripped of all these organelles. So it doesn't have a nucleus. So there's not much DNA in a red blood cell. You would see there's no protein manufacturing, meaning it doesn't have a Golgi apparatus, it doesn't have endoplasmic reticulum. Again, things that would allow our red blood cell to assimilate to a very small shape. And then most importantly, it doesn't have any mitochondria. So since it lacks mitochondria, and we know where is most of the bulk of ATP being produced? In the mitochondria. Right? Okay. We know that the mitochondria was involved in the Krebs cycle. And we saw the electron transport chain where we're able to produce a bulk of all of our ATP. So therefore, if you were to take a look at that red blood cell, how do you think it would produce ATP? If it doesn't have mitochondria, how does it produce ATP? One portion that doesn't require oxygen.
Fermentation.
Fermentation is a step before that.
That's
The first step of cellular respiration.
anaerobic.
What do you do with glucose?
Glycolysis.
Glycolysis. You split it. Glucose, glycolysis. So glycolysis produces what? How much ATP?
2.
2 ATP per glucose molecule, right? So if you were to measure the enzymes in a red blood cell, they would be all glycolytic enzymes. These would be enzymes that are involved in able to churn out that ATP. So technically, our red blood cells can not only get energy from lactic acid, from glucose, but they can also get it from lactic acid as well. Another thing about the mitochondria, the red blood cell, is that it doesn't actually use any of the oxygen that it carries. So because we know that the oxygen was heavily involved in mitochondria, it was an oxygen or an electron acceptor. And at the end of the electron transport chain, we saw oxygen picking up those high energy electrons and combining that with oxygen, a split oxygen molecule, we produced water. So therefore, since red blood cells actually don't use any of the oxygen, it's able to carry it. So that makes sense that our red blood cells would want to be delivering oxygen and not using it, so it's technically not using its own supply. Another thing too about the red blood cells, another thing about our red blood cells is that we have a tendency to remove them after they're no longer usable after 120 days. We have macrophages in the liver that will come in and destroy that red blood cell. It'll start to chop it up, and it'll use those amino acids, it'll use the contents of the cell to make new red blood cells. Looking at the dysfunction of red blood cells, this is where you would see sickle cell disease, a disease in which the misshapen red blood cells, the deformities that are present in red blood cells due to a genetic disorder, reduces the oxygen carrying capacity of red blood cells. It's called sickle cell shaped red blood cells that no longer have that semi-donut shape and therefore can't carry oxygen as much. So people with sickle cell disease do seem to have respiratory problems simply because they're not capable of carrying oxygen. They can do normal activities, but as time goes on, they find that they can't perform heavier activities due to the reduced oxygen carrying capacity. Another thing is leukemia. Leukemia is the presence of immature red blood cells. So like I said earlier, these stem cells that are present in the bone marrow start to become erythroid stem cells. And at some point, erythrocytes, the red blood cells that are produced, will eventually turn off the production of red blood cells, again, which we call erythropoiesis. But people with leukemia will note that there is no regulation in stopping the production of erythroid stem cells, so they find that the red blood cells are constantly producing red blood cells. So leukemia is known as what's called a liquid tumor because it's a tumor not of any mass anywhere in particular, but that of the blood itself. Now, within the red blood cells, we have hemoglobin. Okay? So don't think that hemoglobin is a red blood cell. Don't get that confused. We have, zooming into the molecular structure, it is a protein of which we have 4 subunits that are put together to create this hemoglobin molecule. And the whole sole purpose of hemoglobin is to carry oxygen to every tissue in the blood that the capillaries are travel through. And so we have about 300 million molecules of hemoglobin present per red blood cell. So naturally we want enough of these to be able to carry them. So not only is it carrying oxygen, but it also carries CO2. So we'll learn about this at the very tail end of our cardiovascular system lecture. And how does hemoglobin carry and how does it release oxygen? How does it carry CO2? And then also what is involved in producing our bicarbonate buffer. So red blood cells and the hemoglobin specifically serve that function. If we were to take a look at the subunits, like I mentioned earlier, there's 4 subunits of which there's 2 types, our alphas and our betas that are put together in order to carry it. We'll have 4 heme groups, 1, 2, 3, 4, and its capabilities of carrying oxygen as well. White blood cells we won't talk too much of, but nonetheless, white blood cells can be divided by granulocytes and agranulocytes. It's the presence of these granules in the white blood cells that serve as, these granules serve as a lytic effect, basically the ability to lyse a foreign cell. So if there's something that doesn't belong in your bloodstream, these cells have the capability of going in and destroying that cell. So what it's looking for specifically are cells that do not belong. We're looking for things based on the cell markers, things that are present on the outside of the cell that our own immune system can recognize. And I did talk a little bit about a couple of the autoimmune disorders, mainly like type 1 diabetes. I did talk about Hashimoto's disease. They're called autoimmune disorders because our own immune system don't recognize those cells. So they think that it is a foreign agent, and these immune cells come in and start attacking those cells and destroying them over time. Another reason why those that have transplant surgery, if anybody gets a heart transplant, kidney transplant, they have to take a lifetime supply of what are called immunosuppressants to suppress the immune system from attacking tissues that are considered foreign. And we can't just take anybody else's kidney, we can't take anybody else's heart, we gotta find somebody that's a very close match that matches the markers on top of each cell. And I'll talk a little bit more of those markers later on. Platelets are going to serve the function of blood clotting, and they work together with autocrine and paracrine signals. For the purposes of being able to aggregate around an injury. So take a vessel and nick the vessel at some point, and we want to have platelets that clot around that area in order to prevent any blood leaking from that point. So these platelets actually utilize ATP, so ATP to ADP. Not something you really need to know, but the phosphorylation of ADP forms a process in which it starts to stimulate other platelets to come stick to one another. So in other words, in order to enhance the stickiness, you're going to see that platelets use ATP in order to start formulating this clot. If you look at the structure of platelets, you'll see again, very small. Here you can see all the red blood cells scattered around, and all these dots are going to be our platelets. So no nucleus, really very limited number of cellular organelles, but mostly cytoplasm from other stem cells. So as I mentioned earlier, looking at where these are originate from, stem cells, these are the remnants of them. So they are very specific platelets, or very special function for what they do. The other thing about platelets also, because they do exhibit their stickiness, another thing that I don't think we'll really touch on it, is what we call atherosclerosis. This is the form of plaque forming on the side of the wall. So take a fatty diet, for instance, and you start to form these clumps that form around a vessel. And those clumps that form together create turbulence as that blood flows through. And any turbulence in that area starts to accumulate platelets around that area that will start to form this plaque wall around. And what that's going to start doing is you're going to start doing something called stenosis, where it's going to start to narrow the pathway that blood can follow. So here you start to see the passage where blood can flow through start to get smaller and smaller. So that starts to reduce blood flow into that area. So that would have prominent impact on any high functioning vessels or high functioning areas such as the heart, where any plaques that form that can come in and dislodge and block that area can start to reduce the ability for our own circulatory system to deliver blood to that area. So this is a condition we call ischemia. The lack of blood flow to an area that can result in the death of cells. So if you were to look at myocardial ischemia, is reduced blood flow to certain areas of the heart. The next thing is to take a look at the blood groups. So on top of the red blood cells, how our own body recognizes our own red blood cells is the presence of surface markers on the cells themselves, which we call antigens. So antigen, if you're ever to read any information on with antigens regarding the surface markers, you see that cells have glycoproteins. These are proteins with little antenna on top. So these glycoproteins with these huge carbohydrate chains right on top are markers for the cells to let them know that these are our own cells, not to attack them because we own these cells. So these antigens are present also on our red blood cells, of which we have two types of antigens that are specific to us. What I'm trying to get down to is really your own blood type. How do they characterize your blood type? So the way this is divided is we have two specific antigens. We have the ABO blood group, and then we have an RH antigen. And starting off with the ABO blood group, you'll see that we have two main alleles which are the types for a gene, would be A and B. And these are specific markers on top. And you can see on the image on our bottom here, you can see A antigen would be expressed with a purple dot, while the B antigen would be expressed with this arrow, this light blue arrow. And so, while it may not look exactly like this kind of gives you the idea that antigens are specific too. So we do express these, and I'll talk about our ABO blood group in just a little bit. The RH antigen is another antigen that's present, of which either you're positive for it or you're negative for it. And most people have these. About 85% of the population has the RH antigen, so they're positive for RH. And the importance behind this is whether you're positive or negative when we talk about our blood groups. So RH antigen is going to be another thing that's also profound in pregnancies, because and we don't get too much into it, but if you look at pregnancies, women that are pregnant can be negative. They can have like a negative, they're negative for the RH antigen, but the fetus can be positive. So that would mean they receive that positive antigen from the father. And so that positive, that RH positive baby living within an RH negative female, we know that blood can't cross between the two, so that won't compromise itself. So the fetal blood can maintain separate from the maternal blood. But if there's ever any injury on the fetus of any kind, or any on the placenta or anything like that, that causes fetal blood to leak out into the maternal blood, the mother starts to produce antibodies against that RH antigen. It knows how to attack for it. So that has an impact on future babies for that pregnant woman. Simply because if they ever have a RH positive baby, there's antibodies that would start coming in and starting to attack it, because it doesn't belong. The other portion, again, the ABO blood group, as I mentioned, there's two different antigens, which are A and B, which I pointed out earlier. And from what you were gathering, what I'm talking about is that the antigens are expressed both from the males and females. So we undergo meiosis for the production of sperm and eggs. That combined, we're going to get half the DNA from the mother and half the DNA from the father. So we know that with our antigens, with conception, we're going to see the maternal DNA and paternal DNA come together. And so we can start to take a look at the concept of genotypes and phenotypes. Now, genotypes are what you carry, and phenotypes are what are expressed. So to give you an example would be would be black hair, or the gene for black hair, as well as the gene for blonde hair. And we know, I'm thinking of Game of Thrones while I think about this. It would be, black hair would be a dominant trait. So while a carrier, the genotype would be having both black and blonde hair, the genes for it, the one that's expressed, the phenotype, would be black hair. So we'll start to see how that takes place, of which we're going to have two of each allele, simply because one belongs from the mother, the other one belongs from the father. So with that, here you can see our genotypes. Let me write this somewhere else. So genotype down here, and our phenotype over here. So our genotype in this case, meaning will be a carrier for those alleles. So someone who would be type A, type B, type AB, and type O. That's the expressed phenotype. What would be their genotype? Meaning somebody who is type A can either have AA or AO. O being the recessive allele and A being the expressed allele. Hence, therefore, the phenotype would be A. So if you're a genotype AA, you would be blood type A. Somebody who's type B would have a genotype of BB or BO. Somebody who is type AB would actually express both antigens, AB, meaning they've gotten the A from either the mother or the father, and the B from the mother or father. And then type O would express no antigens, meaning they would have to be OO. So here, type O is not going to have any markers for the AB antigen, but they still will express RH antigen, either they're positive or they're negative. That we didn't include on this particular slide. So with that, if we're to take a look at the most common type, about 39% of the population is O positive, while AB negative is the rarest type, of which we have 0.5%. So like I said, blood type expression. We're going to start to look at how, when we take a look at the male's genotype as well as the female's genotype for the ABO blood group. The male would have to have his own genotype. In this case, we can make him AO. So this, in that case, his phenotype would be type A. And the female could be type B. So again, we're using the example. Even though they're type B, they're either BB or BO. But in this case, I'm just showing you BO and AO. So here you can see the different combinations based off these two alleles, where the male exhibits AO, A and then O, and the female is B and O. So with the different combinations of what this could be, that A allele can go to A and combine that with the O and then the O and the B, give you type AO, AB, BO, and OO. So how does that play out? How do we figure that out? We use what's called a Punnett square. Have you guys seen these before?
Yes.
So Punnett squares are fairly easy, in which all you're really doing is taking the allele of the male and the allele of the female, and you're putting them together. So we get AB, we get BO, AO, and OO. And you'll see that these are the different combinations of these two. So from these, now you can see the genotypes of the offspring. So we have basically a 1 in 4 chance of any of these blood types taking place. But the phenotype that's expressed would be AB, type B, type A, or type O. Those are the phenotypes expressed, of which you have, for AB, we have what's called codominance. Meaning that both A and B are expressed. A doesn't trump B, nor does B trump A. The one that is trumped is the O allele. So when we see BO, we see type B. Same with AO as well. And then the last one, to be type O, would mean you'd have to get the O allele and the O allele from the male and female. So with that, AB being codominant, this doesn't count for the Rh antigen. If we were to do a Punnett square with our Rh antigen, that would have to give us a 4x4 square. And that makes things a little bit more complicated, or we get about 16 combinations of. But for simplicity's sake, we can just look at this and then we can account for the Rh antigen later. Blessings. So now, based off this information, let's take a look at matching donors and recipients. So any compatibility for our donors, which are our columns on top, and our recipients, which are the rows across, the ones that are compatible are going to have the check mark with them. So here you can see anybody that is negative, Rh negative, for our recipients, cannot receive any blood from anybody that is Rh positive. If they were to have Rh blood, let's say you get a blood transfusion of the wrong type of blood, what would happen? Well, your own immune system cells would come in and start to attack those red blood cells. They'll start to clump together. And as they clump together, they'll start to clump even more, and eventually we'll start to clot and reject that blood. So obviously not a good thing, hence why they want to make sure that your blood types match. So all of our negative for our Rh antigen can only really receive blood from those that are Rh negative. The other thing to take a look at too is those that are AB positive, recipients that are AB positive, can receive blood from anybody that's A, has type A blood, type B blood. They can receive blood from anybody who has positive or even negative antigen. So here they're compatible with everybody. So in this case, because they can receive blood from O positive, O negative, B positive, B negative, they're called universal recipients. So these are the lucky ones that can receive blood from any blood type for both antigens. Now conversely, here you can see O negative. And since O negative doesn't express any Rh antigen, nor does it express any A or B antigens, they can donate blood that looks just like this. So therefore, they can donate to any of the blood types. O negative can donate to O negative. And here you can see O negative can only receive blood from O negative, but O negative can donate to every single type. Therefore, they are called the universal donors. And if you're an O negative, is anybody here O negative? You probably have you guys ever donated blood before? Do you get a lot of calls? Yeah, all the time, because you're a universal donor. So that's all it comes down to. Is anybody here AB positive? I've donated blood like how many times I still don't know my blood type, but nonetheless, any way you look at it, if you're an O negative, which my mom was, calls all the time. I remember had to take her calls because it was always the blood donation center calling out and wanting her to donate. And I forget, how many weeks can you donate?
I think it's every 30 days.
Every 30 days? Better believe they're calling you for more blood. So that is, being O negative, you are the universal donor. We are coming up with ways, in fact, April 2007, they came out with a way to strip the antigen completely off the blood itself. And even though that was about 10 years ago, I don't know where the progress has been made, if we're still able to do this, but there is what's called a glycosidase enzyme that comes in and can cleave those antigens off. So if you can take of any blood type, whether you're type AB, and then run it through with this enzyme, and there it comes off and chops off all these antigens on top, well now effectively what you have is type O blood. So with that, you would be able to create a universal donor type blood if you can strip the antigens. Because really all you need are the red blood cells. You need the hemoglobin contained within the red blood cells in order to boost your oxygen carrying capacity. So if you're ever in need of blood and you're low because you've got low red blood cell count, you want to make sure you can get that hematocrit back up. You want to make, or if you're anemic, you want to make sure that you can get your red blood cell count back up in order to be able to deliver the amount of oxygen that you need in order to sustain life. So if they can strip those off, well, then they can work with that. Another thing too that's interesting about that type of blood is that they've actually created artificial blood as well, where they have stem cells, just regular stem cells that they can start producing red blood cells without any markers. And they know what they were trying to do is mass produce red blood cells. If we can start producing red blood cells, then we shouldn't really have as much of an issue with blood donations. Because most of the time it's really just rejection that takes place. Yes?
I'm curious, and I know I'm just missing something. And they ask if you're HIV positive when you donate blood. There's no cell nucleus and no mitochondria. Where is the HIV hiding?
So what's where is it hiding? HIV positive. Specifically, it's going to be, and I don't know that much about HIV, but it's not just the red blood cells. It's specifically the T cells that have been modified. So,
where is that in the red blood?
so red blood cells, if you were to take a look at the makeup of them,
Yeah.
red blood cells are going to be the bulk of it. So it's not affecting the red blood cells, it's the white blood cells. So what happens with HIV positive, just like any virus, what's really cool, viruses are really cool. So if any of you guys ever study viruses, we're starting to learn to use viruses to inject DNA that we actually want. Because what viruses do, they come in, they attach to a cell, and they literally will penetrate that cell and inject its own DNA into it. So what happens with any of the viruses, and whether you're looking at cold viruses, flu viruses, they do the same thing. They come in, it takes a host cell, and in this case that would be the white blood cells, or the T cell, a specific white blood cell called T cells, where the HIV comes in, injects its own viral DNA into it, and reprograms that T cell to start producing more HIV viruses. And every time it produces another HIV virus, the DNA that's contained within that virus starts to mutate just a little bit. There's some mutation, just like you guys learned in that lab that we did, the slight point mutations that occur that change the nature of the HIV. It's still HIV, it still will affect you, but they can never really sequence HIV to say, okay, maybe we can get rid of it. Well, if the virus mutates over and over and over again, they're always constantly playing catch up with the DNA sequence for that particular virus. Hence why when you look at flu vaccines, why haven't we been able to eliminate the flu vaccine? It's because every time it jumps from a person to person, it's going to mutate, and that mutation is going to create a new type of flu virus. And we see all the things that are coming out, H1N1, all derivatives of the flu virus that come back out that will affect us and start to affect our immunity. So what's happening with the white blood cells is they start to come in, they create antibodies for that type of cell, and now you can fight it off. So typically, if you've ever had a flu before, you might not catch that flu again because you have the antibodies stored in. But to answer your question, that's all in the white blood cells. That's where the DNA comes in. All right, so lastly, for blood, we'll look at blood lab results. And here is an old printout of our blood labs. Let me just zoom in real quick. Some things to point out. The reference ranges. Reference ranges are the expected ranges for your demographic that are produced. So you can see from reference ranges represent the homeostatic balances for each of these. Here you can see a chemistry panel, and I want to just divvy this up for you, where you can see these would be things that are dissolved in the blood. So this is a measure of the plasma, while these are going to be a measure of the formed elements. So naturally, you'll be able to see glucose levels, and again, the reference ranges of where your glucose levels should be, 136 to 144, and that's really determining whether this person's fasting or not. You look at sodium, potassium, chloride. These three from our chemistry panel are just measuring the electrolytes in our blood. Again, we want to make sure that we maintain normal values of that, because what happens when any of that is excess? I did ask you that quiz question of, was it a quiz question? I think it was something related with potassium. What happens with excess potassium in the extracellular fluid? So naturally with that, any excess in potassium can start to affect our nerve cells from firing, the excitability of cells, and upset the balance of those. Glucose, chloride, carbon dioxide are gases, creatinine, look at cholesterol levels. Here they can look at the metabolic panel. They can look at the proteins that are present in blood. Do we have enough albumin, bilirubin, which is a byproduct from the breakdown of our red blood cells, and so forth. Very bottom, we'll show you our form counts. So this is our what we call our CBC, a complete blood count. And what they're really doing is looking for the counts of your white blood cell and red blood cells, as well as your platelets. So hematocrit, here you can see 42.3%. That means of that pellet on the bottom that was to settle, we see 42.3% of that entire blood sample is what we call our hematocrit, of which most of that is going to be our red blood cells. Here they can do the amount of hemoglobin, the grams of hemoglobin per deciliter of blood. They can count the number of red blood cells, count the number of white blood cells. They can count also your platelets from that test. And then your differential is looking specifically at your own immunity. It's looking at your basophils, your monophils, your lymphocytes, and everything else. We want to make sure that those counts are normal. Because if your white blood cell counts are high, it means you're fighting off something. We want to look at those specifically. So with that, let's take a look at the heart. You guys want to take a break for a little bit before we go on? I have maybe about 20 slides. I'm going to breeze through it because most of it's anatomy. Yeah? No? 5-minute break? 5-minute break. Okay, great. Let's do a 5-minute break.